Hello and welcome to class today. Today we're going to talk about tissues. Mainly, we're trying to get ready to learn how to identify them. We want to know what their functions are and where do we find tissues. First of all, let's start out with the different types of tissues that we will be looking at. And the types of tissues that you'll find in your body are epithelial tissue, connective tissue, muscle tissue, and nerve tissue. So all the tissues in your body will fall under one of these four categories. Of course, your nervous tissue is made up of your nervous system, brain, spinal cord, and all the nerves coming out are going to be nervous tissue. Muscle tissue, there are three types of muscle in our bodies. Skeletal muscle, which we think of as voluntary, but it's actually innervated by the somatic nervous system because we can control that. Then we have cardiac muscle and smooth muscle, which are both innervated by the autonomic nervous system. When we learn about the nervous system at the end of the semester, we'll think about autonomic in terms of automatic, things that happen automatically that you don't have to think about, they just do. We also will have epithelial tissue and connective tissue, which is going to be some of the major types of tissue that we're going to be looking at. Epithelial tissue is basically, you can think of this type of tissue, it is going to cover and line everything in your body. So it covers and lines. The specific functions are forming boundaries, protection, secretion, absorption, and filtering. So when you have epithelial tissue is primarily involved. Your skin, known as the epidermis, is actually, or the epidermis portion of your skin is made up of epithelial tissue. And of course also it lines all the organs, and we'll talk a little bit about that. And then connective tissue. Connective tissue functions include supporting, protecting, and it is the tissue that binds all other tissues, which is why we call it connective. It connects. Your bones are a type of connective tissue. Your tendons, fat, and soft padding tissue, that's going to be connective tissue. Another one, blood. You probably haven't thought about blood, but blood is actually a connective tissue. We can add to those functions transportation, because blood is, and that's mainly the tissue for transportation, is your blood. The substances are transported through your body in your cardiovascular system, or circulatory system, through your blood. All right, we're going to begin with epithelial tissue. Epithelial tissue is, because it is going to cover and line everything, that means it's going to have a free surface. So we call that having polarity, which means there's a free surface. It's apical. We refer to it as apical. So anytime you see the terminology apical surface, it means it's the free surface. And then we have a basal surface of epithelial tissue, which is where those cells attach to other tissues. And remember, all epithelial tissue is going to have a basement membrane that connects to connective tissue, because connective tissue is the connecting tissue of our body. Oftentimes these apical surfaces, remember apical meaning the free side. If you think about all epithelial tissue is going to have a free side, so if you're looking in a microscope, you're going to look to the edge of a specimen, typically. But very often, we will find on these surfaces microvilli or cilia. Microvilli are there to increase surface area. For example, when we are absorbing nutrients in our digestive system, we need an increased surface area. So these little microvilli make, there's more areas for them to absorb, to come in contact with those cells. Cilia, they look very similar to microvilli, but they're smaller structures. They act like paddles. For example, in your respiratory system, breathing in, you don't want to breathe in all these little particles in the air. There's somebody outside mowing right now. If you walk through that, you're going to breathe through some dust. Well, the cilia will trap the dust out, of course, of your respiratory tract. So that's that nasty stuff you either cough up or blow out of your nose, and the cilia are catching it and moving it out like a paddle. They have ATP that pushes them one direction, so it'll move all that stuff out. Now, one of the things that will help you identify epithelial tissue is that this type of tissue is tightly packed. It's cell to cell to cell to cell to cell. When you see, it's going to be very different from connective tissue, because connective tissue has a lot of extracellular matrix, so it's not as many cells butted up next to each other. So when you see a bunch of cells together, you're probably looking at epithelial tissue. There will be an exception, as the adipose tissue is the adipose tissue actually looks kind of like it's cell to cell, but actually the adipose tissue is just squished with fat, so it makes it look like cell. You can't see the extracellular matrix, and I'll show you that when we get to that picture. So it's tightly packed cells, a continuous sheet, and they're connected to each other by tight junctions or desmosomes. And between these cells is usually where you're going to have filtration or absorption. So those functions are able to be met through the way these cells are connected to each other. Another thing important to remember is that epithelial tissue does not have a blood supply. That directly, the blood supply does not come directly into the epithelial tissue. But epithelial tissue is able to be nourished those junctions in between the cells. So the nutrients filter out of the blood vessels and filter on into the epithelial tissue because of the nature of this tissue. And the other thing to note about it is a high rate of regeneration. When the cells that divide very quickly to regenerate this type of tissue. All right, when we're dealing with epithelial tissue, the first thing we want to do to identify it, first of all, we said look for a bunch of cells together. So looking at the picture, we see cell to cell to cell to cell. This is more of a cartoon depiction. I'll show you some real tissues momentarily. But first thing you want to do is ask, is it simple or stratified? Simple means it's a single layer. Now, when you're looking at a microscope slide, this can be a little confusing. I'm going to try to teach you a little tricks so that you can identify this, but it depends on whether you're looking at a cross-section or a longitudinal section as to whether you're looking at simple epithelium or stratified epithelium, and I'll show you, I'll give you some pointers to help you in that identification process. But the first one, of course, you see the simple, just a single layer, and then at the bottom is stratified, so that means more than a single layer. So 2 or more layers of the cell is going to give you a stratified. So we always categorize epithelial tissue as either simple or stratified. Then we also classify epithelial tissue according to its shape. We have 3 different shapes. And when we're identifying the shape, you look at the apical layer of cells for the stratified version. What does that mean? I'm going to look at the very top, the free surface, right? Remember, apical means the free surface. So I'm going to look at the layer of cells there to be able to identify, because as we get closer to the basement membrane, sometimes it can be very confusing. So we want to look at the shapes of the cells on the apical layer. They can be one of 3 types. They can either be squamous, cuboidal, or columnar. Now, squamous cells typically look kind of flattened out like a pancake. Look at the nucleus also. So when you're identifying the cells, you can look at the outer boundary of the cell as well as the nucleus. So looking at squamous, we kind of have a pancake-shaped nucleus and a pancake-shaped cell with a hexagonal shape. Sometimes this hexagonal shape is not absolutely obvious. Now, the cuboidal cell underneath it is real boxy looking, and the hexagonal shape on these cells is, you can't see it very much in the microscope. But it's a very boxy cell, and so is the nucleus. The nucleus is nice and plump, very boxy. Then we look at columnar tissue. It's an elongated cell. It looks like a column, so we call it columnar. And the nucleus is also elongated, so that helps us in identification of this tissue. And also on columnar, and sometimes we'll have what we call a pseudo-stratified columnar tissue, and we'll look at that in a second. But often you'll find cilia or the microvilli on these columnar-shaped cells. So looking at simple squamous. Now, the picture that you see here is a little deceiving. It's not my favorite picture to use because simple squamous, right here, this picture here, in the lung tissue. If you were looking at other types of membrane, it would actually have more of that hexagonal appearance. Kind of looks like fish scales, or or sometimes a little different variation, but you can see it. So if you see a slide, you kind of have to just kind of look at this slide and say that is definitely lung tissue, because that's the only way the simple squamous tissue will look like the picture that you see here. In your TAL, which I study in my Mastering AMP course, you will see a better picture of simple squamous tissue. But where do we find this type of tissue? We find it in the kidney glomeruli, which is a structure we'll talk about when we cover the urinary system in AMP2. But we need this type of tissue because it allows the passage of materials through diffusion and filtration, and your kidney glomeruli are the filtering apparatus. So that's where blood filtration actually occurs in the glomerulus of the kidney. And so it makes sense that we would have simple squamous tissue. So remember, a single layer of simple squamous tissue definitely got to have a lot of filtration, things moving back and forth, because it's going to be a thin layer that allows that to happen. And remember, the junctions between those cells, or the desmosomes, allows for that passage. You'll find it in what we call the endothelium of vessels. When we cover blood vessels, we'll talk about the endothelium, which is always going to be simple squamous tissue. That's because, again, filtration and absorption has to occur. And we also find this type of tissue in the mesothelium. And I believe in PAL, the example is mesothelium of the serous membranes, and you can see a little bit better of a picture than we have here in the PowerPoint. The next type is simple cuboidal. And I'll give you a little hint. Very rarely do you see this type of tissue in the stratified version. It does come stratified, but mainly simple cuboidal. And look how boxy those cells are. And look how big and fat the nucleus is in each cell. And remember, we're looking at the apical surface. Now, because this is a cross-section, what you're seeing is little kidney tubules, so that you can see the center of those. So the center is like a lumen, because those are tubules. So this would actually be considered the apical surface. So here's one here, here's one here, here's one here, 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 here. So you can see several little tubules, and they're lined with the simple cuboidal epithelial tissue. So we're looking at that real boxy shape. Remember, real boxy and a big fat nucleus. Where do you find this type of tissue? Of course, the kidney tubules, as we see, ducts and secretory portions of small glands, so where secretion occurs around glands, so they would line a gland, and the ovary surface also has this type of tissue. All right, and let's look at the columnar. Remember, these are elongated cells, and you can see the very much column shape here. Now, these nuclei are a little boxier than normal, okay, but we can obviously see that it's columnar because of the elongation of the boundary of the cell. A lot of times you'll see the nucleus will be more like this, kind of shaped, elongated as well. But in this particular picture, they're a little more boxy than usual. This is a single layer, of course. It is simple. And we actually find this particular type of tissue in non-ciliated types will line most of your digestive tract. Remember, the digestive tract, it really doesn't need cilia, because cilia move one direction, kind of trying to get rid of something, and weenus does all the way through the digestive tract, so they're non-ciliated. You also have this type of tissue in the gallbladder. Some of your ducts and glands will have this type of tissue. You'll have a variety of this that's ciliated, remember, in the respiratory tract, so in the bronchi. Then you'll have some simple columnar cells or tissue in the uterine tubes and some regions of the uterus. Pseudostratified columnar epithelial tissue. Sometimes this can be very confusing when looking, so you're going to have to kind of study these pictures on PAL. The way that you know that you're looking at pseudo-stratified, meaning it looks like multiple layers, but it's really not. And it looks that way because the nuclei are not lined up very well. So it looks kind of like multiple layers. But this is pseudo-stratified. The reason is because every cell actually hits the basement membrane down here. So here's the basement membrane. So these cells all are actually meeting it. It just has this appearance. Something worth pointing out here is a goblet cell. And here is your goblet cell. They're usually kind of whitish and they're shaped like a goblet. This is an important cell because it secretes mucus. So goblet cells secrete mucus, and you'll find them usually in the pseudostratified columnar tissue. So anywhere you need secretion and propulsion of mucus, you're going to usually find some pseudostratified columnar epithelial tissue. You'll find the non-ciliated type in sperm-carrying ducts and ducts of large glands, and you'll find the ciliated type like that we see here in this picture. So here are the cilia, little hair-like structures that move things usually out of the trachea or the upper respiratory tract. So you'll find that type of tissue in the respiratory tract and in sperm-carrying ducts and large glands. Now this picture shows us stratified squamous epithelial tissue. How do I know, looking at this picture, that is stratified squamous epithelial tissue? Well, remember we need to look at the apical surface. So this is the apical surface here, the free surface, and look at those cells. They're flat cells, flatten out, and the nucleus is real flat. And then I see several layers, so that tells me I'm looking at, and that's the reason why you'll notice that we say look at the apical surface. If you look down in here where we're getting to the basement membrane, those cells begin to look kind of boxy. But we're dealing with stratified squamous epithelial tissue. And that's why here the basal cells do become cuboidal and columnar when we get there to the basement membrane. But these cells are all squamous, and they're stratified. The function here is to protect the underlying tissues, and usually you'll find stratified squamous tissue in areas of high abrasion. There's also a non-keratinized form lining the esophagus, mouth, and vagina. Remember, these are areas that are subject to lots of abrasion. And you'll have a version of this that's keratinized, where you'll be able to see the keratin in this type of tissue in your epidermis. And keratin is a special protein that keeps our skin from drying out. So remember, it has to protect. Our skin is a very major portion of our protection of our body. It protects everything inside. So it has to be able to hold water, not let water escape. So keratin in the skin does so. We have some rare types of epithelial tissue. Stratified cuboidal, like I was telling you earlier, is pretty rare, but we do find some stratified version, but it's usually only a couple of layers thick. And then stratified columnar is extremely rare as well. There's a little bit in the pharynx, there's a little bit in the male urethra, lining a few little glandular ducts, and you might find this at a transition area between types of epithelia. A lot of times you'll see several types of epithelial tissue. This type of tissue here is called transitional epithelial tissue. How do I know that I'm looking at transitional? I look at the apical surface, and the cells have a balloon appearance. These cells that have the balloon appearance, they're called umbrella cells. And they're a little bit stretchy. They can stretch out a little bit, and they're transitional because they look like they're transitioning between different types of epithelial tissue. The basal layer, again, you'll see some columnar, some cuboidal. But this dome shape or balloon shape on the apical layer is what helps you to identify the tissue. And where will you find this? Ureters and urinary bladder. These are both areas of stretching and distension. Now, we find epithelial tissue in our glands. The endocrine glands and the exocrine glands are all lined with epithelial tissue, because remember, epithelial tissue lines and covers everything, and a duct kind of is going to be lined. It's going to secrete something. And the difference between an endocrine gland and an exocrine gland is what's actually happening. And an endocrine gland actually secretes hormones. And that means it's going to secrete these substances into the blood. A hormone is any substance that's secreted into the blood that's not acting locally. And an exocrine gland is a more immediate effect. It secretes like sweat, for example. These are more numerous than endocrine glands, and these secretions are typically released on the body's surfaces. And so good examples are mucus glands, sweat, oil, and salivary glands. So these are all different types of exocrine glands. Now I'm going to stop right there for now and pick up. I'm going to pick up momentarily with another video for you to finish out talking about tissues. You'll hear from me shortly.